Hey everyone, take a deep breath and settle in. Today, we are going to tackle a comprehensive, incredibly detailed review of oxidative phosphorylation. Consider me your personal, very patient tutor for this explainer. If you're currently grinding away, preparing for your university biochemistry exams, you are in exactly the right place. We're going to meticulously break down this final, highly energetic stage of cellular respiration, so you can walk into that test with total confidence. Here is our mental roadmap for today's session. First, an introduction to oxidative phosphorylation. Then, we'll look at the mitochondria and shuttles. Next, electron carriers, followed by complex 1 and ubiquinone. After that, complex 2 and alternative pathways. And finally, we'll wrap up with complex 3 and the infamous Q cycle.
All right, let's kick things off with Section 1, Introduction to Oxidative Phosphorylation.
So, let's look at the big picture. Up to this point in your metabolism studies, you've likely spent hours agonizing over glycolysis and the Krebs cycle. You've watched how those earlier pathways break down our dietary carbohydrates and fats. But crucially, and nota bene, note this well, all those oxidation steps are accompanied by the reduction of electron carriers. We're constantly reducing NAD+ to NADH and FAD to FADH2. Essentially, all of those preceding metabolic pathways are really just funnels. They're funneling high-energy electrons right into this final phase, stage 3. This is where the central process of energy metabolism truly hits its peak.
Moving right along to Section 2, the mitochondria and shuttles. Let's zoom in on the anatomy of our cellular power plant. The inner mitochondrial membrane is strictly where the magic happens. Notice how this inner membrane folds into crista to massively increase its surface area. Now, this membrane is highly impermeable, which, as we'll see, is completely intentional. Anchored right into this inner membrane are our main electron transporters, complexes I through IV. Meanwhile, the matrix inside houses your Krebs cycle enzymes, and that intermembrane space, that serves as a holding pen for the protons we'll be pumping. One quick thing to mentally flag here. Cytochrome C is uniquely water-soluble, meaning it can move freely rather than being locked down in that lipid membrane. Now, here's a massive functional problem the cell has to solve. Cytosolic NADH, the stuff produced during glycolysis out in the cytoplasm, simply can't cross that impermeable inner membrane. There's no door for it. So the cell uses a brilliant workaround, called the malate aspartate shuttle. The enzyme malate dehydrogenase effectively takes the electron equivalents from NADH and uses them to reduce oxaloacetate into malate. Malate actually does have a VIP pass to cross the membrane into the matrix. Once it's safely inside, another malate dehydrogenase grabs it and reoxidizes it back to oxaloacetate, yielding a fresh, brand new molecule of NADH exactly where we need it.
For your exams, I want you to strictly associate this shuttle with kidney, liver, and especially cardiac tissues. Think about the human heart. It requires an absolutely immense, continuous supply of ATP to just keep beating, right? There are no breaks. This shuttle ensures that the electron assembly line receives an uninterrupted, constant flow of cytosolic NADH equivalents, completely bypassing that membrane's impermeability. Without this specific indirect transfer mechanism, cardiac muscle literally could not sustain its non-stop energy demands. It's a lifesaver.
Let's move to Section 3, Electron Carriers.
So what actually carries these electrons? Cytochromes. These are heme proteins, meaning they contain a prosthetic group with an iron atom right at the center. It's exactly like the hemoglobin in your blood. That iron atom alternates between the iron 2+ and iron 3+ states to carry electrons. We can clearly contrast their specific structures here. Cytochromes A and B have their heme groups bound non-covalently. But look closely at heme C. The heme group here is covalently linked to the protein via thioether bonds, specifically formed by cysteine residues. That structural difference is huge.
Look, if there's one table I highly recommend you commit to memory for your exam, make it this one. It's high yield. You'll see that cytochromes A and B are fixed solidly in the inner mitochondrial membrane, held there by those non-covalent bonds. Cytochrome C, on the other hand, is the odd one out. Thanks to those covalent thioether bonds we just talked about, it is a fully water-soluble molecule. This means it can physically detach and swim freely through the aqueous environment of the inner membrane space to deliver its electrons. It's the mobile courier of the system.
Negative 219 kilojoules per mole. I want you to strongly anchor this number in your mind. This is the massive amount of standard free energy released when electrons travel all the way from NADH down to oxygen. Now, if the cell released all this energy at once, in a single massive reaction, most of it would just be lost as uncapturable heat. By breaking the electron transfer into multiple, smaller redox stages across the different protein complexes, the cell minimizes thermodynamic loss and efficiently captures this energy to pump protons. Let's actually look at how this builds mathematically, because professors love to ask you to prove this on exams. We use the standard Nernst equation framework. The standard free energy change, delta G prime naught, equals negative n, which is our number of electrons, so that's 2, times Faraday's constant, times the change in standard reduction potential. By taking the O2/H2O semicouple at the cathode and subtracting the NAD+/NADH semicouple at the anode, you get a potential difference of 1.136 volts. Plug all that in, and the math proves exactly why this overall process is so highly exergonic, mathematically yielding our negative 219 kilojoules per mole.
On to section 4, complex I and ubiquinone.
All right, let's meet the big one. Complex I, officially known as NADH ubiquinone oxidoreductase, is an absolute giant. It's this massive L-shaped structure made up of 42 distinct subunits. Look at the clear structural division here. The horizontal membrane arm is deeply embedded in the lipid bilayer. That's the part responsible for the actual physical pumping of protons. Then you have the vertical matrix arm protruding straight down into the aqueous matrix. This matrix arm is exactly where our NADH docks to begin the entire sequence.
For the exam, understanding this exact step-by-step path is absolutely crucial. First, NADH binds to the matrix arm and hands off its 2 electrons to a cofactor called flavin mononucleotide, or FMN, reducing it to FNH2. From there, the electrons don't just jump randomly, they are passed systematically down a highly organized conductive wire made of iron-sulfur clusters. Finally, these electrons are delivered to their final destination within the complex, a molecule of ubiquinone, which you probably know better as coenzyme Q.
Let's look really closely at coenzyme Q itself. On the left, we have fully oxidized ubiquinone. Notice the benzoquinone ring and that incredibly long tail made of isoprenic units. That long hydrocarbon tail makes the molecule intensely hydrophobic. And that's not just a fun fact, it's a functional necessity. Because it's so hydrophobic, once it accepts those 2 electrons and 2 protons to become the fully reduced ubiquinol you see on the right, it can easily detach from complex I and just swim laterally straight through the oily lipid membrane over to complex 3. Now, a massive word of warning for your exam. Ubiquinone does not accept both electrons at the exact same time. It is a strictly stepwise process. Accepting just 1 electron and 1 proton turns it into a semiquinone radical, QH dot. This is a highly reactive intermediate state. It absolutely must accept a second electron and proton to finally become the stable ubiquinol. Do not skip over this intermediate radical state in your studying. Test writers love to see if you know it.
Section 5, complex II and alternative pathways.
You are almost guaranteed to see a compare and contrast question on this, so let's carefully map it out. Complex I uses NADH and actively pumps 4 protons across the membrane. Complex II is fundamentally different. First, it uses FADH2, not NADH. But most importantly, Complex II does absolutely 0 proton pumping. None. It contributes electrons to the chain, sure, but it does not contribute directly to the proton gradient itself. Furthermore, it's actually a dual-purpose enzyme. It is the exact same succinate dehydrogenase enzyme that you already memorized for the Krebs cycle.
Taking a microscopic look at complex II, you can see it's built from 4 distinct subunits, A, B, C, and D. Subunit A is the catalytic part sitting right in the matrix that converts succinate to fumarate. Covalently bound FAD accepts the electrons, becoming FADH2. The electrons then travel through the iron-sulfur centers located in subunit B. Finally, they reach subunit C and D, which are the integral membrane proteins containing a type B heme, where they are successfully handed off to another waiting molecule of ubiquinone.
And finally, section 6, complex 3 and the Q cycle. So, our fully reduced ubiquinol from both complexes I and II is now swimming through the membrane toward its next destination. Complex 3, or cytochrome BC1, is a massive dimer with 22 subunits, but here we hit a strict biological speed limit. Ubiquinol arrives carrying 2 electrons, but cytochrome C, that water-soluble carrier waiting on the other side, can only accept 1 electron at a time. To solve this bottleneck, complex 3 uses the intricate Q cycle.
Ubiquinol binds to the QP site and gives up both electrons. One successfully goes to cytochrome C. The other electron is deliberately diverted to the QN site to partially reduce a waiting ubiquinone back into a semiquinone radical. This cyclical recycling is absolutely crucial for continuous flux. Also, jot this down. The iron-sulfur centers here are Rieske variants. They uniquely use histidine residues instead of cysteines to coordinate the iron.
Let's do the final math on our proton gradient, because it's incredibly satisfying. When a pair of electrons starts at NADH and travels all the way down to the final electron acceptor, oxygen, at complex 4, we get a very precise tally. Complex I physically ejects 4 protons. Complex 3 ejects another 4 protons. And finally, complex 4 pumps 2 protons. That gives us a strict, undeniable total of exactly 10 protons pumped from the matrix into the intermembrane space for every single molecule of NADH.
So look at what we've accomplished. We have successfully transformed the chemical energy of NADH into a highly concentrated electrochemical gradient. An entire reservoir of positive charge is now trapped in the intermembrane space, like a coiled spring. Which brings us to the ultimate question that we will tackle in your next rigorous study session. How exactly do these 10 precisely pumped protons flow back into the matrix to physically rotate the mechanical motor of the ATP synthase, forging the very ATP that powers life itself? Keep that amazing mechanism in mind as you review your notes today. You've got this exam in the bag. Happy studying.